Here, we're going to talk about how ATP is made and how ATP can be used. We're talking about this in the context of skeletal muscle cells, more properly known as muscle fibers. Remember, muscle fibers have lots and lots and lots of energy generating stuff in them. They have muscle fibers have lots of mitochondria, the powerhouse of the cell, which makes ATP. It makes ATP from sugar. Muscle fibers have lots of glycosomes, little vesicular organelles that carry glycogen, which is really just packed up, bricked down sugars. They break down very easily into the simple sugars that mitochondria need to make ATP. Muscle fibers also have lots of myoglobin, which is like hemoglobin. Remember, hemoglobin in the blood, it's what makes your blood red. It's what makes your red blood cells red. They are quaternary protein structures, which means they're just like a bunch of individual protein structures bound up to one another to make one bigger, more functional protein thing. They're quaternary structures, quaternary proteins that have iron molecules embedded within them. Iron attracts oxygen very, very happily. They attract one another very, very happily. When hemoglobin attracts that oxygen to its iron, it rusts like your rims on your car, or my car anyway, and it rusts and it turns red, right? These are your oxygen carrying molecules in your blood. Your muscle fibers have a protein that's very similar to hemoglobin, but are unique to them. There's some modifications that also hangs on to a lot of oxygen. And what do you need that oxygen for? To fuel ATP generation in mitochondria. So glycosomes have stuff that helps fuel the mitochondria. Myoglobin has stuff that fuels the mitochondria, and the mitochondria can use those things to make ATP. Now we're going to talk a little bit about glycolysis and the electron transport chain and the Krebs cycle. We're not going to go in tremendous amounts of detail here. I do expect that when you learned about this in introductory biology, that you did in fact learn it and you remember a fair bit about it. So these slides are going to gloss over the key points that you really need to keep in mind when you're thinking about muscle contraction. But I assume that this is a refresher and that you know more than this. Okay? So we mentioned ATP is the main energy storage unit to do work in the cell, basically any cell. Specifically in a muscle, you need ATP to move the myosin heads and help form and release cross bridges. So you need ATP to bind. See, there's your myosin head. You need ATP. There's your A and your 3 Ps to bind to the myosin head to cock it back in a ready position. When one of those pops off, it releases energy. This is bound to an actin binding site. It'll be pulled this way. That energy released will pull the myosin head, and that'll drag the actin filament along with it, thereby moving the actin filament closer to the M line. And of course, this is happening over and over and over and over again. That AD will pop off. Your myosin head will release from the actin binding site, and then a new ATP will come in and recock that myosin head in a ready formation. So ATP will get, in its original form, will get the myosin head ready for the cross bridge formation. The energy release when you pop that phosphate off of ATP to make ADP releases energy, and that energy is used to pull the myosin head forward. ATP also opens a calcium pump in the sarcoplasmic reticulum. Remember, you need calcium inside the myofibril in order to facilitate contraction. Calcium binds to troponin. Troponin is bound to tropomyosin. Tropomyosin covers the actin binding sites. So when calcium comes in and binds to troponin, it changes troponin's shape. It pushes the tropomyosin out of the way, and those actin binding sites are revealed so that they are capable of binding with myosin. So ATP moves the myosin heads physically in the first place. They also open a calcium pump that lets calcium in to do the actin side of this transaction. And it also returns sodium and potassium back to a starting gradient after excitation-contraction coupling. Excitation-contraction coupling will come in a different set of slides. You'll need to refer to those for the details about this. But basically, your ATP is involved in moving myosin heads around, shuttling calcium to where it needs to be, shuttling sodium and potassium to where they need to be. In any given muscle cell, despite having all of those glycosomes and all of those myoglobin, you still only have about 4 to 6, so like 5-ish seconds worth of ATP in the muscle cell. So you need to be constantly churning out ATP. Just having a bunch of glycosomes there in the first place and myoglobin to make one batch of ATP isn't enough. You need to be constantly able to replenish your ATP. You're going to need to keep breathing, you're going to need to keep eating and getting those nutrients into your muscles to facilitate contraction. If any part of this falls apart, contraction starts to fall apart, slow down, weaken, etc. So when ATP, we say it's the main energy storage unit for work in the cell. When ATP is it dehydration synthesis or hydrolysis that'll happen here? Hydrolysis. Remember, lysis means breaking down. When ATP is hydrolyzed into ADP and a phosphate ion, it releases a bunch of energy. And that's what's going to do the work. It's the release of that energy when ATP is broken into ADP and phosphate that'll do that work. ATP has to get right up next to whatever it is that needs to be moved in order for that energy to be harvested by that thing. So you have a lot of ATP and you have it a lot of different places in the cell, and you need to keep the cell full of this ATP to be able to fuel its various functions. ATP can be replaced quickly in 3 different ways. Direct phosphorylation, popping that P back onto ADP to make ATP. It can be replenished by the anaerobic pathway, or ideally, the aerobic pathway. Aerobe refers to oxygen. An refers to without. So the aerobic pathway uses and requires oxygen, which you're going to hang on to with your myoglobin, right? Anaerobic does not need, or sometimes cannot happen, particularly when you talk about anaerobic bacteria. They really need for there not to be any oxygen around to work. Does not need oxygen. And direct phosphorylation is just, you're just popping that P back onto the ADP pretty directly without a bunch of any other energy generating steps. So kind of the most straightforward way of those 3 processes, direct phosphorylation, I always mispronounce that word, anaerobic or aerobic respiration is direct phosphorylation. It's the simplest one. It just regenerates what you already had. It doesn't make new ATPs, but at least restores what you did have, so that that ATP isn't just gone forever. You have a molecule in your muscle cells called creatine phosphate, which is also a high energy molecule, and this is stored in muscles. So it's sort of ATP-ish, but you're not going to really burn it directly. Instead, so you've gone through the whole mitochondria stuff or glycolysis or whatever, and you've made your ATP. And then you use your ATP, pop off a phosphate, boom, release of energy, and now you have ADP. Creatine phosphate can come along, pop its phosphate onto the ADP. Now you've got ATP again. Pretty straightforward. You're just remaking the ATP you've already used. This does not create more ATP. This phosphate that's loosey-goosey in the cell can go back and help replenish creatine phosphate again. So this is sort of its own self-perpetuating cycle. Now, if things break down, things get worn out. So, there is entropy in the cell as well as the rest of the universe. So this isn't something that can be sustained infinitely, but it can help keep you going in a pinch. The anaerobic pathway is basically just the first step of aerobic respiration, but it kind of ends there. So if you recall, aerobic respiration, you have glycolysis, where you have 1 6-carbon sugar breaks down into 2 3-carbon sugars, pyruvate. Each of those can go through the citric acid cycle or the Krebs cycle. I tend to prefer citric acid cycle, but tomato, tomato, it's all the same thing. Glycolysis makes a little bit of ATP. Citric acid cycle makes a little bit of ATP, but a lot of a different molecule. The next step is in the mitochondria, where you remember all those enzymes in a row, or those, you have the little pump, and here's your plasma membrane inside of the mitochondria. You have your electron transport chain. And this makes lots of ATP using some of the byproducts of the citric acid cycle. So glycolysis is some ATP, some ATP plus electron transport chain fuel. Electron transport chain makes, as a byproduct, and other steps do too, H2O. This makes CO2 as a byproduct, and lots of ATP. Glycolysis, more specifically, makes 2 ATPs. Citric acid cycle makes 2 ATPs, and the electron transport chain makes 34 to 36 ATPs. So if you do the whole oxygen-fueled stuff, because all this requires oxygen, you end up with 36 to 38 ATPs. But since all that big sort of hugely ATP generating stuff requires oxygen, you can still make ATP, just a lot more slowly by sticking with this step. So the anaerobic pathway uses glucose and engages in glycolysis, splitting 1 glucose into 2 pyruvates. Or pyruvic acid, same thing. You spend 2 ATPs to start the process. You make 4 ATPs, so really you end up with net 2 ATPs. This process happens in the cytoplasm of the cell. This happens in every cell, okay? This is the first step of oxidative phosphorylation. It's the first step in ATP making. It's going to happen in every single cell, including a muscle fiber. So this happens inside of the cytoplasm. The oxygen requiring stuff happens in the mitochondria. In this case, you've made a little bit of ATP, you've got your pyruvates. Now what? If there's not oxygen to use those pyruvates to make more ATP, your body will break down that pyruvate into lactic acid. The buildup of this lactic acid is what makes your muscles burn. You know, you have that sort of like that actual like burny feeling when you're really working your muscles, and the soreness you experience the next day has a lot to do with the lactic acid buildup in the muscle cells, in the muscle fibers. Now, when I was taking AMP for the first time, they said that was why you had the muscle soreness, like period. It was just all about the lactic acid. Turns out to be way more complicated than that, but it is a factor. It is still a factor. This lactic acid will migrate out of the muscle fiber and into the bloodstream. From there, lactic acid can actually be used by other organs as an energy source. Your muscles can't use it as an energy source, but other stuff can. Your liver, for example, your kidneys, your heart. Your liver, in fact, can reconvert that lactic acid back into glucose to feed back into this process, which is a pretty cool trick. It's a bit of a slow turnaround because it has to go through your muscle all the way through your liver, and then your liver has to do stuff and has to send it all the way back to your muscles. But it's still pretty cool. It can still regenerate it pretty nicely that way. So this doesn't make much ATP, right? It only net 2 ATPs, but look how few steps there are. You break down that glucose, and there's a series of enzymatic reactions, but they're all super fast. You break down that glucose into 2 pyruvic acids, you've released your ATP, done. End of story. In the muscle, at least. That lactic acid will migrate out. So it's short. It doesn't make much ATP, but since it's short, you can do it a lot faster. You can start the process over and over and over again really fast, provided you have enough glucose in the first place and ATP to fuel the initial breakdown of the glucose in the first place. So it's about 2.5 times faster than the aerobic pathway, which I think actually speaks to some pretty cool stuff about the rest of the aerobic pathway, because this stuff is like complicated and there's a million steps in there. And to make it so that glycolysis is only 2.5 times faster than the whole rest of this super complicated machine really speaks to me to the efficiency and elegance of the rest of that process. It's super highly conserved in nature. Basically everything that lives does this trick, maybe with a handful of tweaks here and there, but it's basically the same process. It's a very, very, very efficient, elegant system to make energy. So assuming you have a normal amount of glycogen in the cell, a normal amount of glucose in that muscle fiber, you have enough of this to keep up with your ATP needs by itself for about a minute. If you had no oxygen whatsoever, none, and you've depleted whatever existed in the muscle fiber in the first place, you could keep making ATP this way for about a minute. Okay? This is not inherently sustainable either. So direct phosphorylation is just sort of this snake eating its tail, this self-regenerating beast that eventually will break down because entropy is a thing. But it will maintain itself for a while. The anaerobic pathway can go on to make more energy than what was put in, but only for a little bit. Pretty quickly, which is nice. Very few steps, which is nice. It makes a byproduct that can help fuel other stuff, which is nice, but it is pretty limited. The aerobic pathway is what we just talked about with all the extra stuff, all the extra cool stuff that we all hate to memorize because it's complicated. You have glycolysis, which is the anaerobic part. It is not oxygen. It does not require oxygen. It can happen. It's the first step of this whole process, so it can happen with oxygen around. It's just it doesn't need any. Citric acid cycle, aka the Krebs cycle, oxidative phosphorylation, the electron transport chain, all require oxygen to happen. So these are aerobic processes. Requires O2, anaerobic doesn't care either way about whether or not there's oxygen around. So, same process in the first part. Glycolysis is the same. You put in 4 ATPs, you end up with two net ATPs. You spend 2, you make 4. 4 minus 2 is 2. The pyruvic acid moves into the mitochondria, goes through the citric acid cycle where you make 2 more ATPs total. That citric acid cycle, if you recall, goes around twice. You make 1 ATP per cycle, 1 ATP per pyruvate, because you have those 2 pyruvates, so that's why it goes through twice. You make 1 ATP per pyruvate, so the citric acid cycle per glucose makes 2 ATPs. The electron transport chain, which happens in a different part of the mitochondrion, makes 32 to 34 more ATPs. It is a crazy efficient powerhouse of a system. Boom, boom, boom. This process can be sustained for a very, very long time. As long as you have oxygen coming in, as long as you're in an aqueous environment, you can keep this going, and your mitochondria work, you can kind of keep this going as long as there's, and glucose, stuff to feed the system. And a lot of this stuff can regenerate itself. These enzymes can sort of get reset back as part of the process. So this can happen for a very, very long time with very little degradation. It does take a little longer, but again, compared to the anaerobic pathway, not crazy long. This is all a really elegant, efficient process. There's alternate fuels that can be fed into this. Glycolysis, it's glycolysis. Glyco refers to glucose. You need glucose to do this thing. The aerobic pathway, you can have a bunch of alternate start points that are fed by different molecules. So you can use protein, for example, to feed this process. That protein has to go through other steps to get to where it can be fed into the citric acid cycle, but it goes straight to that step. And it does burn more energy to get to that step, so it doesn't quite yield the same amount of energy. It's a little bit more expensive, but you can do it. That's one of the other beauties of this process is you don't need straight glucose to make this happen. Fats, can be broken down eventually and be fed into this process too. The major byproduct, aside from ATP, of aerobic respiration is CO2 and H2O, carbon dioxide and water. And because it makes so much ATP, even though it takes a little longer and uses more stuff, it is by far the preferred pathway of the cell. About 95% of your ATP is made this way. Pretty cool, huh?